everything we just talked about as far as the action potential, right? Starting from a local area over here, right? You're going to get that depolarization that's going to spread to the next area over here, right? And then it's going to spread to the next area. And we knew it spread in this direction just because you have those, so the potassium channels behind it, repolarizing it, right? But otherwise it goes in one direction. It's going to go from one segment to the next segment, to the next segment, to the next segment, right? Just walking down like that stack of dominoes. That's called continuous conduction. And this is how it would go if you didn't have so-called myelin. Right? So that was continuous conduction, everything we've talked about, right? Ions are flowing through their voltage gated channels in each individual segment by segment, and just a random kind of localized depolarization. The next one is with myelin, right? This insulation layer, and it's going to result in so-called saltatory conduction. Right? This is when you have a map action potential traveling down an axon that has been myelinated. Myelin is going to provide this insulation, and we're going to see this potential, right? This fluctuation of nerve jump from these regions in between the myelin sheaths. This is called saltatory conduction. This is the whole purpose of myelination. So back to our glial cell stuff, we had oligodendrocytes in the central nervous system. Their axons came out, right? And they would spread along different segments of a particular axon, right? And this particular axon and this axon, right? So one oligodendrocyte might go out and insulate a small section of a bunch of different axons. And between each segment, there's a little space in between right here. There's an oligodendrocyte. In your peripheral nervous system, you had a so-called Schwann cell. Each Schwann cell is going to myelinate a particular segment, and only that segment. It's not going to reach out and get another one, right? So if the axon's the hot dog, your Schwann cell is the hot dog roll, right? Covering little segments right here. This is just myelination. They're doing the same thing. They're just doing it. One's in the central nervous system, one's in the periphery. They're doing it a little differently. And I should say they have different sort of characteristics to them. So certain autoimmune diseases, for instance, might attack oligodendrocytes, but not Schwann cells, and vice versa. So they are very different, even though they're doing different things, right? They have different sort of molecular signatures, as we say, right? So you can get things, different types of demyelination disease, as we'll talk about a little bit, in your oligodendrocytes versus your Schwann cells. And that'll be out in your nerves or inside your central nervous system. So when we talk about myelination and demyelination, especially for your central nervous system, this, again, we talked about gray matter and white matter, right? On the outside, over here, you have all your cell bodies and dendrites over here. And in these neurons over here, they're talking to each other, right? This neuron's talking to this one. This one is maybe sending his axon out, but he's sending out axons and talking to this guy and this guy and this guy. Right? Everybody's just bouncing around signals. That's what they're doing in here. Ultimately, though, they're going to pass that signal on to another neuron, right? And so it's all going to relate to myelination. So you have neurons, cell bodies, right, over here, and then they might be talking to each other with these unmyelinated axons, passing on signals like this, Now I'm talking to him, and these would be unmyelinated, right? But at some point, you may want to talk to someone over here very fast, or you maybe want to talk to someone down your spinal cord, or you maybe want to talk to someone on the other side of the brain, right? So these are for long distance stuff, right? These myelinated axons, because we're going to have to, these can be slow enough, right? That you don't need that. They're small enough. It's a short distance. You don't need that myelination. But here, this might be a motor neuron that is telling you to, cause an action. It's got to get down to your spinal cord very fast. So it's going to be myelinated right after it leaves, right, the gray matter. Okay, so this will be this portion over here. And then, this over here. Right? And then these will all be starting to get myelinated at this point. And the term for that in the central nervous system, we'll talk about tracks, ascending, descending tracks. That's just myelinated axons, passing signals long distance, very fast. So that is just one of those things which will come up a little bit later. I wanted to kind of bring that up when we're talking about myelination. The big point here is we want to pass signals very fast from one place to another. The other place we'll see that myelination is in your nerves. In fact, your nerves are pretty much nothing but myelinated axons. And so here's the axon, so-called nerve fibers. They have this myelin sheath around each individual one. And your nerves are just bundles, large bundles of these axons coming from the periphery, going out to the periphery. Sensory neurons, motor neurons going out, sensory neurons coming in. Right, so these are just myelinated axons, passing signals because this neuron over here is in your spinal cord, as we saw last time when they were very long, right? This is going to be over here, passing over here, and then it's got to go all the way down to your leg, right? So it's got to be there very fast. Remember your knee, your reflex, that knee-jerk reflex, right? You tapped it, and it's almost like instantaneous, right? If it wasn't for these myelin sheaths, that's not going to happen, right? All right, so let's look a little bit what that's all about. When you have an action potential traveling down an axon that is myelinated, what happened is this electric current is going to flow through the extracellular fluid, right, surrounding the myelin sheath, and through the site. It's all from one node to the next, right? So you're going to have this depolarization event where sodium is flowing in this area. What does that do? Sodium just flew in, right? A lot of charge over here. If all those sodium, this leaves a blank negative place over here, which means all the positive charge, right? Remember this outside cell was positively charged, is going to move toward that negative spot. At the same time, this positive charge is going to be moving toward the negative, the more negative cell membrane very fast, right? And it gets more negative because these positive charges are moving that way. So you get this whole cycle of positive charge moving backwards over here, creating a negative thing over here, creating this charge. And you don't have to have this segment, and this segment, and this segment with all those voltage gated channels. All the charge is moving within the cytoplasm, as well as the extracellular fluid to move from one node to the next. And that's going to make it much faster. You can imagine, like when I had a live class, what I do, I'd have a row of people, right? Each standing arm's distance away from each other. And you want to get a message to the person in the front of the row, you tap the person right behind you, and you say, hey, tell that person the next person. So each person taps the next person. Whereas this would be one person, 5-person distance in between them, and you just yell to the next person, hey, tell that person, right? It's much faster than tapping each individual one. That makes a lot more sense if you're actually doing it, never mind. In any case, your signal is basically going to leap. Right, start over here. You're going to have this signal over here, and then you're just going to go jump down from node to node, right, rather than each individual segment. Again, the action potential at the first node generates this ionic currents both in the cytosol and the extracellular fluid. Right, positive charges move toward that negative spot that was just created. These positive charges move toward the negative, right? And what that does is, since this all of a sudden becomes more positive over here, that's going to open up that whole voltage gated channel, right, when that happens. Okay, so the resulting ionic flow opens up at the second node and then the third node, right? And so in that way, you're going to get this positive charge leaping from node to node without the need for voltage gated channels, right? So all your voltage gated channels are just sitting in between the nodes right here. That's nice because, there's less sodium coming in and leaving rather than if it was all the way down here. You don't need that ATP ACE pump so much, you're spending less energy. Right? And it's going to be just much faster jumping from node to node. And this is what it looks like when you look at the graph. Why you don't have like these single ones, or you have this blue area is your depolarization event, pretty much going, are the sodium channels rather going from node, opening up from node to node. Each action potential will be measured over here, not in between them. Does that make sense, everybody? So I'll also mention, I think if this is in your slides, again, there's a couple of well-known diseases, Guillain, I think it's spelled like that, Guillain-Barré syndrome and multiple sclerosis. This is happening in your PNS, by the way. This is happening in your CNS. And there's other demyelinating diseases, but you got all this current leakage because your myelin, actually what happens for both of these, probably multiple sclerosis, these aren't genetically passed down. They probably were started off with some kind of virus infection that caused an immune response and caused those immune cells to start attacking the myelin sheath. And then they kind of chewed it away. And then all of a sudden you're left with this bare axon, right, where it should be very fast conduction of speed. You got this big disruption in the transmission of nerves, right, which has caused all sorts of problems in, sensory and motor defects for both of these. So that is these demyelination diseases. That is your myelination speed. Again, you're going from little segment to little segment with your unmyelinated segment. So here is time, right? 1 second. They start off the same. At 5 seconds, this one's further down, right? At 10 seconds, this one's barely moved relatively. This one's almost at the axon, right? 10 milliseconds. It's almost down here due to this leaping, right? So speed is massively increased by these saltatory conduction with the myelin sheath. So if you want very fast conduction, you want a myelin sheath, right? Other things besides the myelin sheath, that's going to vastly increase it. You've insulated it. You've caused these distribution of voltage gated channels. Another thing, which again is kind of hard to describe since we don't go over resistance and stuff, but larger diameter axons, just in general, if you ever run into this kind of material, larger diameter axons are going to be much faster. They're much less resistant. So certain sensory neurons, right, that want to conduct things faster than other things like touch and proprioceptions and stuff like that are going to travel up to the spinal cord much faster because they have a much wider diameter, right? Than other ones, like different things like proprioceptors, mechanos, pain stuff is much slower, right? Whereas these are very fast. And then like the slowest ones are unmyelinated. And as I said, the cooling off, the speed is the temperature difference will also make a difference, right? So that are 3 factors that affect the speed of propagation. All right. So difference between your graded and action potentials. There's a list over here that you can kind of look at the difference of the type, the signal, where is it occurring? What are the gated channels involved? What are the ions involved? And then so on and so on, right? And next time we'll talk about hyperpolarizing signals, because we're going to look at the types of signals that cause that more negative thing. Right? And so that'll be this difference over here. All right, so that's it for this lecture. We talked about the graded potential that was created at the synapse by that initial depolarization event. And if that depolarization event was strong enough, it would reach the threshold of the voltage gated sodium channels at the axon hillock, which would then create an action potential. And from there, the action potential would go in an all or none sequence down the axon toward the axon terminals to release more neurotransmitter at the next neuron. And then the speed of that action potential was greatly affected by the myelin sheath of that in that saltatory conduction. All right, that's it. We'll see you next time.